shouldn't, just so Sierra yells at you. What?
Okay. Is that working? Oh yeah, I guess I need to start. That would have been unfortunate.
Oh, it didn't fall asleep. Nice. It didn't fall asleep.
Well, it's probably
Yeah, it's in presentation mode. Yeah. Oh, because this timer actively is like, yeah.
It's gonna look like you present. All right. Whoa. Oh, sorry about that. Welcome. Thank you all so much for coming to Fishing Friends. It feels like springtime outside. It's really nice. Yeah. No, not. All right. Well, we've got a couple events coming up. We have Science on the Spectrum, which is going to be a webinar online highlighting several people in the STEM field who are on the autism spectrum. So definitely check that out. That's going to be this Thursday at 8 p.m. If you're interested, check out our website or talk to Sierra Barkdahl after this. Not February. That's my fault. Not February. That's March. That's my fault. March. Come on. And next month, also not February, we have a Lukey lionfish collecting and handling workshop. So if you're interested in learning how to safely collect and handle lionfish, join us in Lukey. We'll have a class in Marathon, and then we'll head down to Lukey Reef Resort, where we'll have a free 2-tank dive where you learn how to spear lionfish, and afterwards you'll learn how to collect and handle them. Yes. No, April 28th. April 28th. Yes. There's a lionfish jewelry workshop April 6th if you're interested in getting crafty with lionfish and not going underwater to spear lionfish. That's a great way to get involved with the lionfish invasion. You can learn to create a beautiful pair of lionfish earrings, lionfish necklace, cufflinks, all sorts of really cool stuff. Definitely check that out, and that you can register for that online as well. Our Fish Out of Water 5K, our annual 5K, the registration is now open for that, which is really exciting. I know a lot of you all have been a part of that in past years. So definitely get on the website, pick your fish team. I'm on Team Hammerhead. If anybody wants to join me on Team Hammerhead, they're more than welcome to. There's also Eagle Ray, Emperor Angelfish, and all sorts of other assorted things. And let's see what else we've got coming up. We have an Arts and Science Festival. So if you're interested in coming to the Arts and Science Festival, it's going to be on Saturday, April 29th, 10 a.m. to 3 p.m. It'll be here at the Reef campus. There's going to be artists. There's going to be vendors. There's going to be all sorts of cool things. Madison, who's one of the interns here, spent all day practicing making some really cool craft hats for kids. So if you know anyone who would like to come, or we would definitely love to have you come, join us as we celebrate arts and science as a way to engage with the ocean. All right. And last but not least, our Lionfish Sweeps event is kicking off on April 1st. So as some of you know, in the past, we had 2 Lionfish derbies, one in the spring and one in the fall. And we're moving towards a kind of an ongoing removal effort in our spring. So starting on April 1st, you can drop off lionfish that you spear to the Reef campus. And every 10 lionfish you bring in, you'll be entered in a drawing for some cash prizes, as well as some other assorted goodies. So the first lionfish you drop off, you get one of those cool lionfish neck gaiters buffs that you've been seeing people wear. It's got that lovely design that Jenny is wearing, that rainbow lionfish. So if you want to buff with that on, bring us just one lionfish at least, and you can get that. If you bring in 10 lionfish, you can get a derby rash guard, which the design will be coming very soon. So be ready. It's going to be really nice. It's going to be kind of the style that Lori's got on there, some with the rainbow lionfish and all sorts again. Okay, and then we'll celebrate that together at Sharky's Rockin' the Dock, which is usually the second weekend in July on that Saturday. It's a local event fundraiser that benefits Reef. As soon as they announce the date, we'll let you know about that.
But it'll still be like biggest, most.
Yes, or it won't be biggest and smallest. It'll just be purely for most. And as opposed to in past years where you're doing as a team, this is an individual thing. So you as Andrew Ibarra can bring in 10 lionfish and get a shirt. And then you as Paul Bonatz or Marta Bonatz can bring in 10 lionfish, continuously get a shirt. And we'll be posting the leaderboard both online, outside the Reef campus, and in Sharky's each week. So definitely go for that. And it'll be updated weekly. So don't feel like you can't get on the top of the leaderboard one week. I have a feeling one of the weeks it's going to be like 5. So I believe in you all. Yes, sir.
Is the April 28th part of this? Because it says the events from the 1st to the June 30th.
Yeah.
But this is the 28th is.
Right, the April 28th, the workshop will be something separate. It'll be just kind of an educational event, and those lionfish won't go towards. Yes,
Is that right?
yes, it'll be a 2-tank dive, but it won't be towards the lionfish sweeps. It'll be kind of its own event.
Even though it's April 1st to June 30th.
Right.
The fish we catch during that are donated to REEF for education programs and for tasting events. So the participants won't be keeping those fish.
No, you won't keep. I don't want to keep them, but just they don't count.
Right, they don't count towards this.
Okay.
Yes.
And what time is that again?
It'll start. It'll be all day. It'll start. Yes, it'll start around 8 a.m. and it'll end around 5 p.m. 5:30 p.m. when the boat gets back. All right. And without further ado, it is my pleasure to introduce Andrew Ibarra, who is a monitoring specialist with NOAA and also a former reef intern. So we're very excited to have Andrew here.
Thank you, David. Appreciate it. Hello, everyone. First of all, happy Pi Day. Saw this meme today, so I felt like I had to share it. But today, yes, I'm Andrew Ibarra. I'm a monitoring specialist for the Florida Keys Natural Marine Sanctuary and NOAA, working with the Mission Iconic Reefs initiative. Originally, this talk was supposed to be by Andy Bruckner. He's the research coordinator for the sanctuary, but he wasn't able to come down, so they sent in me, the backup Andrew, to give this presentation. So, yeah, there we go, upgrade. Yes, so I assume many of you have probably heard about Mission Iconic Reefs in the past. Is that right? Raise of hands. Okay, so most of the room. So I don't have to go too far in depth, but Mission Iconic Reefs is a very large, ambitious project, which is bring together a lot of partners to restore the coral reefs of Florida, of the Florida Keys. Focusing specifically on 7 reefs down here in the Keys. It's one of the largest strategies ever proposed in the field of coral restoration. It calls for, 3 million square feet of reef area to be restored, which is equivalent to 52 football fields. And it's also spanning across the timescale of 20 plus years. Working in Key Largo, in the Key Largo office for the sanctuary, I work primarily at Carey's Fort, Horseshoe, and Chica Rocks. I don't work so much down here. So Mission Iconic Reefs is a phased project. Right now, we're looking at 2% coral coverage across the entire Florida Keys. In a lot of areas, it's much less than 2%. Scientists have estimated that historically, coral coverage in Florida Keys for healthy Florida Keys reefs sat around 30% in the past. Our goal, Mission Iconic Reefs goal, is to get that back up to 25% over the next 20 years. And we're going to do that in a multi-phase approach that focuses on different things as we progress over the years. Phase 1A up here, Phase 1A is going to focus primarily on the faster growing species, the acroporids, staghorn and elkhorn coral. These are the corals that practitioners have been outplanting for over a decade already. So with this phase, with these corals, we can start progress immediately. We know how to grow them. We know how they grow. We have a good method for growing them. We have a good method for outplanting them. So it's a really good way to kickstart this restoration progress. Phase 1B, which goes from 2021 to 2028, is going to jump that coral coverage to 15%. Phase 1B also introduces a lot of other species of corals other than elkhorn and staghorn. Phase 1B is going to introduce the brain corals, the boulder corals, pillar corals, star corals, all of those, in an effort to diversify the reef. You can't just have staghorn and elkhorn everywhere. You need the other species for diversification. And those corals right now are in a kind of R&D scale right now. Like there's no great method for growing them. There's no great method for outplanting them because they grow so slowly. So they're still trying to figure out the best way to do this with those species of coral. And that's going to take some time before they get enough stock to outplant. Additionally, Phase 1B is going to introduce grazers back onto the reefs. So Mission Iconic Reefs is not just restoring coral, it's also, quote unquote, restoring the urchins and the crabs, the Caribbean king crabs as well. They're important grazers, they're herbivores. They pick off all the algae and clear the reef so that the reef is not covered in algae, so that it doesn't compete with the coral. And then jumping to phase 2, starting in 2025 and going all the way to 2035, we're trying to bump that coral coverage, live coral coverage, up to 25% on these reefs. And it's also going to focus on and just enhancing all those populations. Here's a diagram showing the different habitats that these 7 iconic reefs have. So you see here, there's shallow reef crest, spur groove, forage tariff. And we have Mission Iconic Reefs has all these numbers and percent coverage goals for each reef and each habitat at each reef. These numbers came from, well, all these corals, every species of corals lives in different habitat. Elkhorn coral doesn't grow in 30, 40, 50 feet of water. You're not really going to see elkhorn down there. They live in the shallow reef crest or the reef crest in like 15 or shallower feet of water. That's where they thrive. Whereas other species like the four reef terrace, deeper water. And so all these percentages just break down what we're kind of trying to aim for restoration goals for Mission Iconic Reefs. So that the reefs can be restored back to more of their natural state. So what's the approach? Mission Iconic Reefs is kind of outlined like there's kind of 5 steps that have been outlined for how this is going to work. The first step starts with site preparation before outplanting. The practitioners already, before outplanting, already clear the reef area of algae, of nuisance species, of anything that's kind of in the way competing with corals. And they do that as they're outplanting, as they're going. Step 2, coral propagation, is the growing of the corals and then the outplanting of corals. Again, already being done by the practitioners. The third step, adding grazers, supplement with grazers. The urchins, spiny sea urchins in the 1980s were basically completely wiped out in the Florida Keys through a disease event. So it really decimated the population to the point of it being functionally extinct. So these grazers, there's a component Mission Iconic Reefs that's growing these urchins, growing these crabs in labs or in the water to be put back out onto the reefs. And you see here, this is little spiny sea urchin being grown in a tank from Josh Patterson. He's a researcher from the University of Florida. He's rearing urchins in a lab setting. And then we have just a general Caribbean king crab photo. But those are the 2 main, sorry, those are the 2 main grazers that Mission Iconic Reefs is targeting. The 4th step is stewardship and maintenance. This, I think, is a really big, is key going down the road, because in order to ensure that these corals survive and thrive, they need a little more help than what they're getting right now. This step kind of means reattaching broken fragments, removing debris, and most importantly, removing fireworms and snails, which have become big predators of the corals, specifically the elkhorn and staghorn corals that are outplanting. And then the 5th step is monitoring and adaptive management. Performance monitoring to collect data to ensure success and failures are documented, and we can use that data to further inform decisions in the future as we go along through this progress, or through this project. So of course, nothing in life is free. This is going to cost a lot of money. It's going to cost a few bucks. In fact, phase 1 is estimated to be just under $100 million. And phase 1 is 5 to 7 years of work. Right now, approximately $25 million has been secured, or $25 million has been user spent so far for the work that's been done. So we just don't have $100 million in the bank. We're consistently funding. A lot of this money comes from federal and state funding and from grants from the Sanctuary, from TNC and NOAA's Coral Reef Conservation Program, as well as money that the practitioners raise on their own. That's all put together in this pool. And then you can see here a breakdown of what each reef is going to cost to restore. Theoretically, it's not an exact science, but you can get the picture. So where are we today? Right now, restoration organizations are outplanting staghorn and elkhorn coral consistently. As of March 2023, the total amount of staghorn corals for Mission Iconic Reefs efforts is a little over 14,000, and elkhorn outplants a little over 9,000 for a total of 23.6 thousand phase 1 outplants. The purple here, or pink, whatever that color is the total amount that phase 1 is aiming to achieve. So far we have 23,000 outplants out of 123,000 out of the goal, which is about 19% of the way there. So where exactly do I come in? So my job, the Florida Keys National Marine Sanctuary and NOAA has developed a monitoring plan for Mission Iconic Reefs to effectively assess the effects that restoration is having on the 7 iconic reefs. This monitoring plan is essentially a 20-year-long research project, and in order to conduct this monitoring, you need a team of people to get in the water and do that monitoring as much as possible. So they hired a field team, a team of monitoring specialists to carry out a variety of monitoring efforts for these reefs. So these are my teammates. 3 of us work in Key Largo, and then the other 3 work in Key West. So we have 2 teams in 2 locations doing all this work. So what, where, how are we doing this monitoring? What exactly are we doing? Okay, let's break it down. So to start, we have each reef broken up into segments. And this is what we're calling Mission Iconic Reefs, what we're calling segments. So these white outlines here is demarcating these segments of reef. So this is Horseshoe Reef. You see 1, 2, 3, 4, 5 segments for Horseshoe Reef. Every segment has what we call permanent monitoring plots that have been established. All these dots, these yellow and red dots, are the permanent monitoring plots. We have restoration monitoring areas, which are the yellow, and control areas, which are the red. Like I mentioned earlier, Mission Iconic Reefs is kind of like a giant science experiment. So we have a variable that we're manipulating, the restoration areas that have corals being put on them, and then we have a control, areas where no outplanting is going to be happening. Each of these dots represent a 10 by 10 meter area. A 10 by 10 meter area of the reef that we will be monitoring for the next 20 years. And so you might ask, how do we know exactly that we're monitoring that same 10 by 10 every single time? Well, we have little pins installed into the reef. These are stainless steel stakes with a Delrin cover. Delrin is just like a type of really sturdy plastic. These covers are kind of screwed onto that stainless steel stake. And we have little tags on them with arbitrary numbers. And we installed these pins into the substrate and epoxy them. This was a really fun part. We did this all last summer. We got to play with these big drills underwater. They're battery-powered underwater drills. They're by a company named Nemo. Highly recommend if you ever need underwater drills. These things are great. Yeah, so we have these pins installed. So each of those little dots that I showed you, 10 by 10 meters, we have one pin set up on the offshore side of the reef, and then we have a second pin 10 meters away from the first one on the inshore side. So they're 10 meters apart. And by having these 2 pins, we can effectively create a square out of them. If you visualize, create a square out of those 2 central pins. And we will know this is the same exact 10 meters every single time that we're monitoring. Yeah, so we install them, we drill them, epoxy them. They look nice and white and pretty at first, but then after a few months, they kind of get like overgrown by algae. So if you see any of these things out on the reef, don't touch them, please. Oh yeah, for sure. That makes my job easier. Yeah, I'd love that. Some of them have a lot of like CCA, calcareous something algae, carbonate algae, which is really tough. I don't recommend doing it with your fingernails or whatever. But if it's like at Chica, where it's like all this fuzzy algae, that comes off so easily. Oops. Okay, so within these permanent monitoring plots, we have 3 main monitoring efforts going on to assess the reef conditions. We have photomosaicing, fish and benthic surveying, and invertebrate night surveys. Since I know the crowd, I know you people like your fish, so we're going to start with the fish surveying. So this past summer, we conducted NCREMP surveying on the 7 iconic reefs. NCREMP stands for the National Coral Reef Monitoring Program. It's a series of survey methods where series of broad spatial snapshot surveying of the reef condition that provides context for local scale studies of tropical reef ecosystems. So basically what that means is we're looking at different habitat types throughout all these different reefs and conducting fish species, composition, density, size surveys, benthic coverage, and coral surveys as well to determine coral density, size, and condition. These surveys happen every 2 years in Florida, and they consist of, again, fish, coral, and benthic surveys. They're also done in the US Virgin Islands, Puerto Rico, the Dry Tortugas, Southeast Florida, and Guam, and then other parts of the Pacific. So this is a very, very broad and global surveying method that is used. Typically, the NCREM sites are allocated throughout the keys through random stratification. But at this time, in addition to all those typical sites, we had an additional 100 sites allocated specifically for the 7 mission iconic reefs, using that same random stratification method. Let's see. Oh, sorry. So we were aboard the MV Mackay, really cool boat, this past August in 2022, going on these NCREM surveys. I was actually the chief scientist for this mission. So I spent like 2 months of my life planning a week of diving, which is crazy, but it was so worth it. It was awesome. Has anyone been on Mackay by chance? No? Okay. So you may have heard of this surveying method before. It's called the RVC method. This is the method of fish surveying that NCREM utilizes. This survey method, so fish are surveyed on fixed points inside of an imaginary 15-meter diameter cylinder. That's so imagine you have these 2 cylinders. You're given a GPS point. You drop down on it, and then 2 divers go out 15 meters each way, and then you're surveying all the fish that enter your cylinder for 15 minutes. Data is collected. You don't just collect fish species and amount. You do fish species, density, and size. So we're also sizing these fish. We're taking measurements to the best of our abilities. We're also collecting benthic topographical data, which basically means we're taking data such as the relief or like the amount of slope that's on the reef, how tall are the sea fans, how tall is the hardest hard bottom structure, things like that. Let's see. Oh, we're taking measurements of the commercially important species, so like all your snappers and groupers and hogfish, lionfish as well. We're measuring those fish. So having done reef surveys before, I will personally say this is not quite as fun as like the roving diver method, but we do get to use these cool little sticks, which I call scuba swords. They're used to help us take measurements of the benthic the benthic and fish as well. But a few of us during the surveying, we ended up having sword fights underwater, which is why we call them scuba swords. And this is how I felt like underwater as I was doing it. So it's a running joke now at this point. But continuing on, so do note that these 100 additional fish survey sites that I'm talking about are not the permanent monitoring stations that I discussed earlier. So these are totally separate. These sites on this map are the random points that NCREM gave us this year for Eastern Dry Rocks, for example. 5 of the 7 Mission Iconic Reef sites were targeted for this 10-day cruise in August. That's Horseshoe, Carys Fort, Sombrero, Lou, and EDR. Chica and Newfound, the vessel, the Mackay, couldn't get that inshore without fear of running aground because those are Chica and Newfound are such shallow inshore patch reefs that we couldn't fit it into the mission plan. So we had to do them afterwards on our own vessels. So the surveying, after diving for 12 hours a day for 9 days in a row, being out at sea, we actually made out pretty well for this mission. It was my first time putting together any sort of like live aboard kind of excursion like this. So I was very, very happy with how we did. Essentially, we did all but 1 site that we were aiming for. These 8 sites that at Carys Fort that we did not do, 8 of these sites that we were given did not have hard bottom. There was sand or seagrass. And with the RVC NCREM method, you don't survey in seagrass or sand. So those were essentially scratched. We only missed one at Horseshoe because of time and weather conditions, so we couldn't get to it. But we completed everything else within 9 days, which was fantastic. So then afterwards, we're trying to go to Chica Rocks in Newfound. And if you've ever been to these sites or know what an inshore patch reef is, this is kind of what you see on some days. The visibility is just awful. We jumped down in there for several days in a row, and we're just seeing soup. It was terrible. We couldn't get much done at Newfound. We only got one site done. Chica, we got 9 and 14, which is pretty respectable. So yeah, the NCREM program managers are still processing and analyzing all the fish surveying data that we collected back in August and for these sites as well. So they're still going through that data. So I don't have any concrete statistics or information to give out to you guys at this point, unfortunately. But the field team, the monitoring team that I'm with, is planning to conduct more RVC surveys at our permanent monitoring plots coming up in early 2024. And then NCREMP is going to be coming back to us to do more of their allocated sites in 2024 as well. So there's a lot of fish surveying that's going to be done by us in 2024, which I'm very excited about. Okay, so the second main, so that was the first main kind of monitoring task that we do. We do benthic and fish surveying for the 7 iconic reefs. The second thing we do is surveying for invertebrates, but we do it at night. So why are we surveying invertebrates at night? Isn't diving easier during the day? Yes, it is easier during the day, but nighttime is when many of the invertebrates are active. You're not going to see many crabs or urchins in the day, especially not anymore with such low population numbers. And remember, Mission Iconic Reefs is more than just restoring coral. It also includes enhancing populations of these critical algal grazers. So the first, I mentioned earlier, the spiny sea urchin and the Caribbean king crab are the two main ones. Caribbean king crab is also commonly known as the spider crab. And I found this meme. I have to give credit to Moat for this Photoshop work. I thought it was hilarious, this little crab. They're called spider crabs or king crabs, but we call them Mithrax spinosystemus. That's the scientific name that I'll be using from now on, probably. And then we have Diadema, aka the spiny sea urchin. These are also being grown out in labs and in the water in nurseries as well. These are photos by a friend of mine, Jim Britson. He's working with Diadema, growing them out in the water. He has collectors that he collects larval urchins out of the water column, and he settles them and grows them out in like little cages. And he contracts Reef Renewal, and he uses these urchins to eat the algae off of the trees and the trays that they grow the coral off of, which is super nice. Okay, so night diving. Surveying for these invertebrates at night was conducted in September of last year off of the NOAA white ship Nancy Foster. This is a massive ship. I've never been on anything like this before. It was like honestly incredibly cool to be on a boat like this. 187 feet long, 40-foot beam, 12-foot draft, and reached top speeds of 10.5 knots. Night diving has never before occurred off of this ship, so like everything was brand new. The Sanctuary has never done night diving like this. The crew of the Nancy Foster has never been involved with a mission like this. So everything was very, very new to everyone. So we had to take a lot of measures to make sure that like we were prepared. There's just another picture. The Nancy Foster is able to carry 3 inflatable ribs for us, which was fantastic. We were able to put multiple dive teams out and send them out to different plots at the same time to survey. So we were doing 3 times the work that we typically, maybe could have done. And this vessel, to prepare for night dives, they brought a crew. So there was only 8 of us, 8 scientists doing these monitoring. They had a crew of 30 people to run this boat for us, which I thought was crazy because they had never done night dives, so they needed like a daytime shift and the nighttime people as well. So it was crazy. And they also have a full gym and movie theater that we use as well. So it was very nice on the boat. Yeah, movie theater on the boat. So the methods, how were we doing the surveying for these invertebrates? So as I said, we're surveying now on our 10 by 10 permanent plots. Basically, 2 divers would descend onto the plot and mow the lawn searching for these invertebrates, for these critters. By mowing the lawn, I mean you just go up, over, back down, over, up, over, down. You just go up and down, searching throughout the reef for these critters. Each diver is responsible for half of the plot. So there's you and your buddy, and you're doing one 10 by 10 meter area. One diver is responsible for half the plot, or 5 square meters worth of area. So if, for example, one of the pins was like here, and then a second pin was at the door, I would survey this half, and then my buddy would survey the other half. And we just go up and down and collect data. We gave ourselves a maximum of 30 minutes search time and a minimum of 10 minutes per survey. Again, for this, since this was a huge boat that will run aground inshore, Chica and Newfound could not be part of the surveying off of this vessel. So we had 5 reefs and 83 plots to survey in 7 nights. The main targets were, of course, Diadema and Spinosystemis, the king crab, but we're also surveying for all species of urchins and crabs, as well as lobsters, conch, octopus, and sea cucumbers. So preparations began at approximately 6 p.m. The diving began 30 minutes after sunset. So at that time, it was like 7:30-ish, 8 o'clock that we were diving. And most nights we were diving from 8 p.m. till 2 a.m. Yeah, it was crazy. I've never done anything like that before. It was very taxing, honestly. Your sleep schedule gets thrown off. You don't know, you don't know what day it is. You really don't know what's going on. You're in your own bubble, and everything's just like weird. But going in, we really did not expect to find much. It was kind of a running joke between me and a friend, where we were spending all this money and all this time to go down and write down a bunch of zeros on a data sheet and then come back. But thankfully, we were pleasantly surprised with how much we actually found and observed. We were able to do basically 1 reef per night, with the exception of Carey's Fort, since Carey's Fort is such a large reef. We consider it 1 reef, but really it's like we break it down by North Carey's Fort and South Carey's Fort. So we spent 2 days at Carey's Fort and 1 day at each of the other reefs. We were able to hit, again, all but, what is that, six plots? The 5 that we missed at Carey's Fort was because of weather. We had a lightning storm that came in on the second night, had to call the night short for safety reasons. But everything else, we got 100% accomplished, well, except for 1, EDR. But we did really, really well. I was very happy with our success. And at the end, we recorded a total of 46 invertebrate species, 15 crab species, 6 urchin species, 8 snail species, 5 lobsters, 2 sea cucumbers. So a decent amount of diversity here that we were finding. So the results, across 5 reefs and a total of 770 square meters of surveyed area, we did only find 25 king crabs, and we only found 65 diadema across 770 square meters of area. So it's very far and few in between. The most common crab that we found is Mithrax hispidus. It looks pretty similar to the king crab. Maybe this is an individual right here of hispidus. But they were the most common crab that we found. And then Gutatus and Argus, this is a spotted lobster and spiny lobster. Spotted lobster, they were all over the place. We were seeing them every plot, basically, multiples in certain areas. I think the highest that I found within 1 10 by 10 was 7 of these spotted lobsters, which is really nice. But they were by far our most abundant species that we were finding. Here, I just included some pictures of the different crabs that we found. I thought it was interesting. Some of them are really pretty, like this one. I forget exactly the name. I think it's the porcelain batwing crab. Not 100% sure. But then we also have the hispidus as well here. This one was a really cool one. Again, I believe I'm not good with my crabs, but it's oscillated swimming crab, I think was this one. And then I have no clue what that one was. This one was Mithrax forceps. This yellow one was like this big, and they're hiding in fire coral. You can see in here, this is blades of fire coral. So you would see a bunch, one here, inside of all these like little colonies of fire coral. And then this one is a king crab that is missing both of its arms and half of its legs. So this poor guy was riding the struggle bus right here. He was crawling along the reef, just like minding his own business. And we saw him. I was like, oh, let's grab him. Because we were also taking measurements. I don't grab things just to grab them. We were taking measurements and sexing these crabs as well as part of this monitoring. So this one's a male, has a phallic-shaped apron, is what it's called, the underside. So we took some photos of him. And then these are some of the urchins that we saw, other than the diademas. This is ventricosus. I don't know the common name, I'm sorry. And then this one's the pencil urchin, slate pencil urchin, I believe. And we saw quite a few of those here and there. And then this is a nightmarish photo of the spotted lobsters. This guy was staring into my soul. He had some beef with me, but then this one's a spiny lobster that was crawling right across our little pin here. So I was like, ooh, photo opportunity, and I snapped a few photos. I thought it was fun. Okay, so now the favorite part of my job is this, the photo mosaics. Just for your reference and knowledge, all these phrases mean the same thing. I'm probably going to use these interchangeably. Photo mosaics, photogrammetry, SFM, and large area imagery, they all mean the same thing. Yeah. So what is photogrammetry? Photogrammetry is the art and science of extracting 3D information and measurements from photographs. The process involves taking overlapping photographs of an object, structure, or space and converting them into 2D or 3D digital models. Key word here, overlapping. Overlap is a huge, huge aspect of photogrammetry. You need a lot of overlap in your images. I know before digital cameras, you had film cameras, and archaeologists used to photogrammetry or create photomosaics of shipwrecks, where they would take on their film cameras different portions of a shipwreck going down it, and then take the film out and they literally physically put them on top of each other to create like one ship, which I thought that was very neat. But now we have more technology coming out that we can use this method, but digitize it. So we can make digital 3D models based out of thousands and thousands of photos that we take. Yes, next. So how does photogrammetry apply to mission iconic reefs and coral restoration in general? We can use this technology to capture entire reefscapes, documenting what the reef looks like at a certain point in time. So there's a temporal and a spatial aspect to this. We can create a historical image of the condition of the reef at this point in time, in 2023, say. 2, we can derive benthic composition statistics from the photomosaic models. Using certain software that's been developed basically specifically for this kind of stuff, we can determine that, for example, from a photomosaic, from a digital 3D model, we can say that this reef is covered by 70% macroalgae, and 20% of it is covered by soft corals or sea fans. 5% of it is covered by sponge, 4% sand, and 1% coral, for example. We can determine all of that. We don't have to do the benthic composition surveying by hand underwater anymore. We can just take a bunch of photos and then the computer does it for us, which is crazy. It's awesome. And the third thing, we can track the success of coral outplanting over time. We can track coral survivorship, mortality, and growth over time, as well as live coral coverage, by revisiting these sites year after year after year, or by imaging them year after year after year, and seeing the progression of these reefs as you put corals out there. And then subsequently, maybe we can start answering questions as to what are the best methods for outplanting corals in these specific habitats. If you outplant elkhorn with genotype XXX, whatever, using this method, is this the best way to put coral out here, or does a different genotype work better with a different method in a different habitat? So we can kind of start keeping track of that a lot better now. But to do this at a large scale for this project, we have over 100 monitoring plots right now. And to do this for 20 plus years, we need a ton of data storage. So breaking it down, our 10 by 10 meter areas require approximately 2,500 to 3,000 photos to create a successful model. That takes about an hour of shooting underwater. We shoot in JPEG and RAW files. So using both of these file formats leads us to 130 gigabytes of data per plot. 130 gigs for an hour's work. That's a lot. So then when you multiply that by 100 plots, we get 13 terabytes worth of data every year. And we're doing this for 20 plus years. So we're going to end up with over 260 terabytes, theoretically, potentially, by the end of 2035. Not to mention, once we create the 3D digital models, those files themselves are approximately 20 gigabytes per plot. So there's a ton of data involved. We're going to need a lot of hard drives. And that is probably the biggest challenge for this project. It's just like data storage. Like, where do we put all this massive amounts of information? Thankfully, we're not doing this alone. The Florida Keys National Marine Sanctuary has partnered with NCOS, the National Centers for Coastal Ocean Science, to help us store data, create the models, and derive the benthic cover statistics. The people who are working at NCOS are also doing projects in the Pacific and Saudi Arabia, doing the same stuff, but at a much larger scale. They're doing 50 by 50 plots and 100 by 100 plots. So they're 5 to 10 times bigger. So they know what they're doing. We get to learn from them, which I'm very glad about. But our data right now is stored in Microsoft Cloud Service called Azure, and it's shared between us and this other, and NCOS. NCOS is another branch of NOAA, by the way. So the sanctuary system is like one branch, and then NCOS is kind of another branch. That's all falls under NOAA. And so we have this cloud service, which we can put a ton of data on it. And then in the office, I have like a 60 terabyte hard drive for physical backups for ourselves. So I'm good for at least a few years until this thing runs out. So how do you actually do this photomosaic thing? So we use certain equipment. We have a DSLR camera. So there's different ways. Like, there's no exact method really to be doing this. So I'm going to describe the way that we are doing it. We have a DSLR camera and underwater housing. We use a 24 millimeter lens and an 8-inch dome port. So this is the camera that we use, as well as the housing. And then photogrammetry involves, so you need scale bars and targets. The scale bars give the computer a scale to work with. So this stick here, right in between, if you see the middle of the two black dots, that's 25 centimeters long. And so with these things sitting within our plots, when you're taking pictures of them and they're put into the computer software, the computer knows this distance is 25 centimeters and it applies it to the whole model. And then these are targets. These weird little patterns are unique on each little thing. The software recognizes these patterns as markers, and we use these to mark the corners and the pins. And the computer software is able to recognize this immediately and just drop a pin on these things and helps us create a square. It'll make more sense in the next slide, I promise. And then we use these little float things to put in the water column as well. When you're taking photomosaics, you lay all this out onto the reef, and you're taking pictures, and you have to stay a certain height above the substrate in order to capture that, full reefscape kind of image. We like to stay 1.5 meters to 2 meters above the reef substrate, and sometimes that's really difficult to do. It's hard to judge like how far off you are. So we were having trouble in the beginning, and we incorporated this neat little idea just to put little floats on these weights. We cut string 1.5 meters long. We drop them. There's this little float that just sits up there, and we can use these as visual cues to keep us at the same level while we're shooting, so that the photos are all at the same level from the substrate. And it helps the computer be able to analyze the imagery and put the scale onto every photo that we give it. Yeah. So this is a data sheet that we use. As I mentioned earlier, we have 2 pins that we installed on each plot. So we have pin 1 and pin 2. These little squares are indicative of the square targets right here. So we put these little squares on the corners of the square, and then the scale bars go in the quadrants. more or less. It doesn't have to be perfect, you know, but there you put 4 within this 10 by 10 meter area. And then we put the floats kind of like on the outside going out here. Yes, so then once the plot's set up, so we lay all this down precisely, you know, we take the bearing from pin to pin, say this is 300 degrees. We take a 90-degree bearing and swim 5 meters out and place the target right here, wherever we end up. And then we go over to the second pin, take the reciprocal bearing from pin 2 to pin 1, and then we take 90 degrees again and then swim out 5 meters and place a target right here. And then we rinse and repeat for the other side as well. So once we get all this stuff laid down, we're ready to start taking photos. The way we take photos is, again, like to mowing the lawn. I mentioned earlier, you just go up and down the reef. So you can see here, this is a kind of a flight path. It's not the best rendition, I guess, but you can see you're going up and down the reef, and then you're going side to side, covering all this area. We set our camera to take 1 photo every second, and it does it automatically. So I don't have to be sitting there pressing the shutter at 1 second at a time. So you can set the camera to do it. You just got to hold it and swim through. That sounds easy, but it's really not that easy, especially when you're fighting current and you can't see more than like 5 feet in front of you. You have no idea which direction you're going, which is why we attach the compass now to the camera, so we're more accurate. But that's more or less how we do this. I mentioned earlier, it takes about average 60 minutes to complete 1 10 by 10 swim through. And then you see here, we have targets. So 1, 2, 3, 4, 5, 6 targets, and then 4 scale bars as well.
How many targets per diver?
Per diver? Oh, there's 2 divers. So 1 diver sets it up, and then the second diver flies the camera.
How fast are you usually swimming?
Pretty slow. Yeah, it's really not fast. The DSLR cameras we have, the Nikon D780, it has pretty high resolution. So we're capturing a lot of overlap, and we have that really good resolution. Well, that's where it comes into play with the computers and stuff. That good resolution. So a lot of people use GoPros to do this as well, which is all fine. They're answering different questions with their GoPros methods because the GoPros don't have that super high resolution for their photos. The DSLR cameras take much nicer photos. And when you put those in a computer, the software sees that and you have a much crisper image. And so then we can go in through the software and we can look at an image, this part of the reef, and say, yeah, that's this species of algae. This is Dictyota, or this is, staghorn coral, or this is octocoral, whatever, blah, blah, blah. The resolution is good enough to pick up on things that are about this big. Let's see, what's next? Okay, yes, so here are my videos. So once we do all that flying, Sierra, video time. Let's see if this works. I think I might have to, this never works. This is what we end up with. This is our product that we come out of with, that the computer spits out of us when we put in 3,000 photos. Let's see, can we play? No good, okay. I'm just gonna go out and do it from here. So yeah, when we take 3,000 photos in the water, put them inside a computer, and it creates this 3D digital thing.
Is it possible to put it into a VR system?
I don't know, maybe, probably. But yeah, so this is a plot at Chica Rocks. If you've ever been to Chica Rocks, Chica is pretty much an outlier for coral coverage in the Keys at this point. There's still a ton of coral at Chica Rocks compared to the other Mission Iconic Reef sites that we have. So this is my favorite model that we have done so far. I don't have the statistics out of this exact model yet, but I would estimate like 20% coral coverage, 25, 30, pushing kind of those areas. You see all these Siderostraeas, all these big brain coral right here, star corals. You have, I I forget the common names, the Monstrea cavernosas, they're just all over this reef. And it's fantastic. And the software does a great job of using these scale bars to create a 3D model. It gives you that depth as well. You can see how rounded the corals are.
Are you seeing stony coral tissue loss disease on some of these?
We are, yeah. We see it in the water. I don't think we've quite captured it in one of these yet, but definitely in the surrounding areas when we're swimming around, we see it.
How come with the healthy coral, it hasn't been affected by the stony coral disease?
No one really knows. I think it could be a lot of factors. I think mainly Chica Rocks is an inshore patch reef, so it's much closer to the coastline than all the other reefs. And for whatever reason, whether it's water flow or something else, these inshore patch reefs, it's not just Chica, there's others across the Keys that are like this, but similar habitats, similar areas where they just have a lot more live coral still. I don't know if they are able to resist the disease better than other corals, or disease doesn't really get there as much. There's really no answer yet. This is another plot also at Chica Rocks. You have a lot of annularis heads here. You just see lots of vibrant color, lots of topography. The model, you can really see the topography out of here. Yeah, so what's it called? The software, or the software basically filters out anything that is moving. So say you take a photo of, like, right here, this chair, one photo of this chair, and then the next photo, a fish swims by. You have a yellowtail snapper right here. And then the third photo, fourth photo, fifth photo are without the yellowtail snapper. The software recognizes that difference and takes out that photo with the snapper, because those other four photos are exactly the same. And it says, this is a different photo. This doesn't work with whatever, with this chair. So it eliminates it.
How many hours does it take the software to complete this rendition?
Like 12, I think, if you have a really good computer, like a supercomputer. This is why we partner with NCOS, because they have all the equipment that's necessary to be able to do this at the scale that we're trying to do this. We don't have the equipment yet to be able to do that. We're trying to get computers situated, but sometimes working with the IT department is tough in the government. They have a lot of rules and restrictions. Oh, totally. There's one somewhere, a scorpion fish was laying right there the entire time. And I'm sure he came out. I did it last week, so I don't have one of these yet for it. It was up at Carey's Fort, but I'm sure that scorpion fish is right there because he didn't move the whole time. So it will be there. This is another one, again, Chica Rocks. This one, you can tell there's still, you see all your coral here, but all this green, these mats and mats and mats of macroalgae covering this whole area. I just wanted to show this as an example of, how you can see what the reef is looking like.
So is it possible to see the cyan if it's on top of the macroalgae? Can you tell if
the algae's growing on top of the cyanobacteria? Yes. Yes. Yeah. We do. Yeah. We're able to do that. We have there's a lot of nuances to it, but the system gives us a list. It shows us a picture, like a spot on this reef, and it tells us this spot right here and asks us, what is it? And there's a whole list of species. And we're able to take so we're looking at this right here, but we're able to take the photo that is attached to that little point of this model. So this model, it's a dense cloud. Each point, so I don't know, I'm describing this really poorly, but this is made up of billions of points. And each point is associated with a photo, one of the photos that I took. And so when you click on a point, any point, it can bring up all those photos that are associated to it. Yes. What do you mean canopy?
Sometimes you have algae on top of an algae.
Oh, that's where it gets tricky. Okay. It gets very tricky. That's when you have to zoom in and really be able to differentiate what is actually growing out of the substrate. Because a lot of times, you'll have a point and you'll have bushels of Dictyota, the wide branching algae. But you know, it has its spot in the substrate, and then it branches out. And so if the point lands on that little branch of the dictyota, that it's not dictyota. It's whatever's underneath that dictyota, whether it's like turf algae or whatever. If it lands right where that dictyota is attached to the substrate, the hold fast. Yes, exactly. Same with sea fans or whatever. If you capture a sea fan that's like folded over the branch of the sea fan, that's not what's on the substrate. So we are able to tell. But to answer your question, when it gets like that nitty gritty, that's when it gets tough. And our partners with NCOS, they are not experimenting anymore, but they use 2 cameras, a dual camera system. We use 24 millimeter lens, which is a little more wide angle. So they have a 24 millimeter one as well, plus a 60 millimeter lens that is able to see closer to the reef. So you're able to get a much closer, sharper image of those finer details. And you're able to use both sets of photos to create this. Yeah, it does rugosity as well. That's what the scale bars are for. And you're able to tell exactly the rugosity of the reef, how tall a coral is, how tall a spur is, whatever. It does all that. And it's like pretty accurate. Very little error. You got a question?
You lose all soft corals because of the current.
We do, yes. Yeah, that's a good point. I was going to touch on that. But yes, the soft corals, especially when you have current or surge, they're swinging back and forth. And sometimes that creates problems because if you're taking photos, sea fan is here, next photo, sea fan's here, next photo, sea fan's here, and the computer it can't recognize that. So then sometimes if there is a very high density of sea fans all across the plot, there will be like holes. There'll be like spaces where you don't see anything. It's just like empty space. And that's because the computer was not able to associate any direct photograph with this point because everything was changing within that photo. It didn't know it was the same photo, same spot on the substrate every single time. Does that make sense?
So you need a sea fan,
Yeah. So there's a lot of AI work that's being brought in. It's very like early R&D, but we're trying to incorporate AI into all of this. That might be a 2030 kind of thing, but that it is an idea. There's people working on that specifically for Mission Iconic Reefs. Then I have one more to show you. So Chica Rocks is very nice and pretty. Here's one from Carey's Fort that's not quite as pretty. This one is mostly just dead coral. It's all gray. There's not much coral. You have like one brain coral right here, I think. Another Siderastrea right there. But I am very impressed with the rugosity question. You see the rugosity, the relief of this reef here. This is probably like 10-ish feet. The bottom's like 28. The tops were like 18 something. So yeah. Yes.
Let me ask you, when you did the survey on the interbrains, and you found 65 diademas, okay, are those diademas being released by the project?
No.
Or they are natural?
Those were natural. Yeah. So far, other than like small scale experimentational releases, there's been no diadema or crab outplanting or like restoration that's happened. Haven't done it. It's still in a restoration or R&D, research and development mode, where researchers, professors, people who are involved with that work are still trying to figure out the best ways to grow them in a tank on land or in the water or whatever. So they're still developing the best practices and scaling up. I think Mission Iconic Reefs says they want 376,000 urchins to be put out on the reef within the next 20 years. That's a lot of urchins. So it's a big goal, and we still need time to be able to get there. Yeah, we do need them. We do. Okay. No, not as much. I don't think it was actually confirmed here. There were some really sketchy looking ones that were like, is this it? Like we're waiting for it, but no, nothing confirmed for Florida Keys. Yeah, thankfully. Okay, let's get back into this. It's already 8 o'clock, but I'll promise. No, I lost. I have to go all the way. This is going to be super quick, but Mission Iconic Reefs, like any good program, incorporates or should incorporate some citizen science work for the public to get involved, to utilize the public to help out. Let's skip that. Okay, maybe you've heard of this already. It's a new program, Iconic Reef Guardians. This is essentially the Mission Iconic Reefs citizen science program, if you'd call it. This is a program that aims to engage divers and snorkelers with Mission Iconic Reefs. So we have the Florida Keys. If you're familiar with Blue Star operators, these are dive shops and operators that are committed to coral conservation. They promote sustainability, good diving practices, and coral conservation as well. Then you have Mission Iconic Reefs, and now we're trying to tie the two things together, or the three things all together, and create a program, Iconic Reef Guardians, where the goal is to empower scuba divers and snorkelers to be knowledgeable and supportive of coral restoration efforts and to become stewards of Mission Iconic Reef sites themselves. So this means, and you can get involved through, there's 3 different tiers. So this program is still in phase, I guess, tier 1. So right now we're only doing this, but right now Florida Keys Dive Center and Quiescence are the only dive shops operating for this program. But you can go out with those 2 dive shops and you can have a passive nursery experience where you get taken out to one of the coral nurseries that one of the practitioners works in, and you can have a tour. You can swim around, experience what the coral trees are like, what's going on. It is important to note that this interaction does not involve touching or working with corals. It is simply like a tour through the nursery. But tier 2, which should be coming hopefully sometime this year, is an active nursery experience where you can get to go in and conduct coral nursery maintenance, whether it be cleaning trees or doing other maintenance stuff that the practitioner needs. And then the third tier, or the third thing that you can do, which we're also ramping up, and which I think is going to be the most important aspect to this whole Iconic Reef Guardians, is the reef experiences, where trained dive professionals lead divers in monitoring and maintenance of Mission Iconic Reef sites. Operators can adopt a site and provide regular maintenance and reporting to support the success of Mission Iconic Reefs. So what this means is that you can do these reef experiences and go out and monitor corals yourselves. And conduct regular maintenance. That means removing snails, removing fireworms, removing those predators that are eating the coral tissue, that are preventing these corals from growing and thriving. There will be this program coming up soon where you can get involved with that, and get your hands dirty and do some of this work. And this is very important going forward, because a lot of the survivability issues that is happening with corals, at least for long-term survivorship, is just they're getting decimated by snails, and they're getting eaten alive by fireworms, and the damselfish are growing algae farms on them. And so it's just like there's so much competing and harming these corals that maintenance on these outplant sites might really be the difference makers. Because there's been small scale studies as well, and that's shown that if you provide regular maintenance, if you get rid of the nuisance species, they thrive better.
How are we going to hear about this when this comes up?
Through the Sanctuary, Mission Iconic Reefs. If you follow anything like that, I could probably let you know, Jane. I'll let you know when that happens. But yeah, I don't have an exact timetable for this, but just keep this in mind if you're ever wanting to get involved. This will be coming in the pipeline pretty soon. This is just a graphic showing all the partners that are going to be involved with the Iconic Reef Guardians program. And then this is a graphic of all the partners that are involved with Mission Iconic Reefs. So dozens of partners. Yes?
My understanding is that there's a lot of groups that are doing coral restoration and that they all share information. Is that right?
Yes, yeah. That's what Mission Iconic Reefs is basically for, to get everyone to work cohesively together and to, tackle this as one unit, basically, instead of having one person doing this, another person doing that, and really, not moving forward as much as we could. And that's it. That's all I got. So sorry, I know I went overtime. My bad.